Hello students, Biomolecule lecture number 3. We are working with enzymes. Now, enzymes, as we said, biocatalyst. You know n number of names of the enzymes and you can now compare role of enzyme with hormones. आपको ये अंतर आपको समझना चाहिए और हमने ये बातें देखी है when we discussed plant growth hormones. Okay, so briefly revising, enzymes are synthesized within the cell. They work within the cell or they are in the vicinity of the cell. Hormones are synthesized at one location. They are transported long distance and worked at distantly located target sites. Enzymes are mainly proteins, very few are RNA or some ribozyme sort of. Hormones may be protein, may be fats. Enzyme action is quick. Hormonal action is delayed. More the enzyme, more the rate of reaction. In case of hormone, the behavior is erratic. More the hormone, more can be the reaction or even it can retard the growth. जैसे हमने ऑक्सिन देखा था. हाइपो कॉन्सेंट्रेशन, हाइपर कॉन्सेंट्रेशन में बहुत अंतर है. More the concentration of auxin is growth retarder, right? Okay, then enzymes are mainly under the control of DNA. DNA ही कंट्रोल करता है क्योंकि मेजॉरिटी आर प्रोटीन्स. लेकिन हार्मोन का बिहेवियर या तो डीएनए कंट्रोल करेगा या फिर कुछ जेनेटिक मटेरियल का वर्किंग हार्मोन कंट्रोल करेगा. So hormone in that way are difficult to discuss. Enzymes are having a specific role. So आपने ये ध्यान में रखना है. Enzyme and hormone. One more thing you are supposed to differentiate is catalyst and enzymes. Okay, ये differences आप लिख के रखो, समझ लो, because it's there, hidden part of your syllabus. Okay, so now we are discussing variety of biocatalyst. Okay, so here I wrote different properties of enzyme जो आप इधर उधर समझ चुके हैं लेकिन इनलिस्ट नहीं किए. So we'll discuss this first and then we'll write down it in a proper language. ये मैंने सिर्फ लास्ट डे नोट जैसे important point लिखे हैं. So first one, they are having 3D conformational. So as you know that linear chain of amino acid polypeptide, प्राइमरी कॉइलिंग, सेकेंडरी कॉइलिंग. प्राइमरी प्रोटीन जो है इट्स नॉट फंक्शनल, इट इज जस्ट अ प्राइमरी स्ट्रक्चर. सो फंक्शनल प्रोटीन बनने के लिए या स्ट्रक्चरल प्रोटीन भी है तो वो प्रोटीन का प्रोसेसिंग, उसको एक पर्टिकुलर स्ट्रक्चर आना बहुत जरूरी है. सो सेकेंडरी टर्शियरी. सो हियर व्हाट इज दैट? प्रोटीन हैज 3-dimensional structure. So it is generally tertiary when working as enzyme. They are having XYZ axis पे वो coiling वाला structure रहेगा. So they are having 3D configuration, 3-dimensional structure रहेगा. It has one or more active sites. एक एंजाइम को एक सबस्ट्रेट के लिए एक से ज्यादा एक्टिव साइट्स हो सकते हैं. एक एंजाइम को एक से ज्यादा एक्टिव साइट हो सकते हैं लेकिन एक एक्टिव साइट एक रॉ मटेरियल के लिए सेंसिटिव होगी. आई रिपीट, लिसन दिस स्टेटमेंट. व्हाट आई सेड, वन एंजाइम हैज मोर देन वन एक्टिव साइट. पर्टिकुलर एक्टिव साइट इज सेंसिटिव टू वन पर्टिकुलर रॉ मटेरियल. बट समटाइम्स इट इज पॉसिबल दैट वन एक्टिव साइट इज वर्किंग विथ few chemicals which are structurally similar. So ये बात हम आगे जाके डिटेल में देखने वाले हैं. Then next one, enzymes are recycled. Whenever enzyme participate in the chemical reaction, enzyme recognizes its own raw material. Then it bind to its active site. It gets bind to its active site. Okay, then there will be processing and you get end product. So raw material is converted to end product in presence of enzyme. Enzyme is released at the end of the chemical reaction. Enzymes are not processed in the chemical reaction. Enzymes set free at the end of chemical reaction. So enzyme chemical reaction में chemical reaction को शुरुआत नहीं कर सकता. Enzyme needs raw material. Rather, to initiate the process chemical reaction, raw material is needed. In presence of enzyme, the rate of the reaction is more. In absence of enzyme, the rate will be drastically slow in some cases. Okay, so enzyme cannot initiate the chemical reaction. Enzymes are not used as a raw material. They are not used in the chemical reaction. They are not processed in the chemical reaction. They just make the process happen. They cause the chemical reaction to happen with faster rate, and at the end of the chemical reaction, enzymes are set free. Hence, they are recycled. Okay, they are specific in action. One enzyme is specific to one particular substrate or one to two, and that to specificity. मतलब एक enzyme अगर A और B chemical के लिए sensitive है, तो कौन से pH, कौन से temperature, कौन से आयन के presence में वो A के साथ काम करेगा, B के साथ शायद वो बदल सकता है. So their action is very specific, very sensitive to pH and temperature, as I said just now. Enzymes require particular temperature, particular pH for working. It causes reversible reaction. Okay, जैसे हमने glycolysis सीखा था. Okay, total phases, glucose to pyruvic acid. Only 2 steps are not reversible. Otherwise, all the enzymes are reversible. Majority of the chemical reactions are reversible, but reversible nature depends on variety of things. Then, reduces energy of activation. You know the word activation energy. A stable raw material won't participate in the chemical reaction. You have to activate it. Once it is activated, some bonds are broken down, then the chemical participate in chemical reaction. So, if enzyme is present, that energy required to activate the chemical is reduced, and hence a stable chemical can participate in chemical reaction. So, energy of activation is reduced in presence of enzyme. So, these are the important points. Okay, so these are there. Along with that, modified difficult language, write down first point. These are mainly tertiary protein. पहला point आप लिखेंगे ना. First point, it has 3D conformation. उसके आगे लिख लो. These are mainly tertiary proteins. लिख लिया. Second point, enzymes do not start chemical reaction. Enzymes do not start chemical reaction. They are not used in chemical reaction. Enzymes do not start chemical reaction, comma. They are not used in chemical reaction. But they increase rate of chemical reaction. But they increase rate of chemical reactions, and enzymes are recycled. And enzymes are recycled. ये process की वजह से enzymes are recycled. Okay, then ये point हम लिख सकते हैं. They have one or more active sites for one particular substrate. One or more active site for one particular substrate, mainly one for एक active site एक या दो raw material के लिए काम करेगी. Okay, then the number of substrate molecule change per minute. Number of substrate molecule change per minute by an enzyme molecule is turnover number. is turnover number comma it varies conditionally. ऑप्टिमम कंडीशन पे टर्नओवर नंबर ज्यादा रहेगा, एडवर्स कंडीशन में टर्नओवर नंबर कम रहेगा. Okay, then they are specific in action. They are specific in action because of critical requirement of minerals, ions, vitamins, presence of mineral, iron, vitamins, temperature, pH. Temperature, pH. Okay, next one. उसके आगे लिख लो, very sensitive to temperature and pH. That is the cause. It is very sensitive to temperature and pH. Next point. All enzyme actions are theoretically reversible. All enzyme actions are theoretically reversible, but depend on energy requirements, availability of reactants / substrate, concentration of substrate or end product. लिख लो, अच्छे से लिख लो. Concentration of substrate or end product, and pH. Reversible means A plus B giving you C. A plus B giving you C. A plus B are raw material, C is the end product. This is forward chemical reaction. C broken down to A plus B is reverse chemical reaction. समझ रहे हो ना? So ये फॉरवर्ड है कि रिवर्स है, ये टेम्परेचर पे डिपेंड करता है, पीएच पे डिपेंडेंट करता है, किसका रॉ मटेरियल का कॉन्सेंट्रेशन ज्यादा है उस पे भी डिपेंड करता है. So it depends on many things. Next one. All enzymatic actions are theoretically reversible, but depends on energy requirement or availability of reactants / substrate, concentration of end products or substrate, pH, etc. लिख लिया? Okay, then enzymes are specific in action. Enzymes are very sensitive to temperature and pH. Thermolabile ये शब्द यूज करेंगे. Because enzymes are sensitive to temperature, they are thermolabile. What is that? See, as we said, protein's structure is important. If you heat it, the disulfide bonds, okay, other linking, other coiling may get disturbed. If the structure get disturbed, obviously the active sites are disturbed. If active sites become non-functional, then the protein become non-functional. This process is called denaturation. If you continue to heat for longer time, proteins are permanently denatured. But if you heat it for a specific period and allow it to cool down, the proteins can be renatured. Okay, मतलब थोड़े देर के लिए उनका स्ट्रक्चरल मॉडिफिकेशन होगा, चेंज होगा, कूल डाउन होने के बाद फिर से वो स्ट्रक्चर रिगेन कर लेंगे. सो दैट इज रीनेचुरेशन. समझ रहे हो? ओके, सो दीज आर द प्रॉपर्टीज ऑफ एंजाइम्स. वैरायटी ऑफ केमिकल रिएक्शंस कॉज्ड बाय एंजाइम. अब आप केमिस्ट्री में बहुत सीख चुके हो, हाउ ग्रुप्स आर ट्रांसफर्ड, हाउ the bonds are broken down, how new bond formation are there. Basically, I think you study two type of chemical reactions. One is bond formation and other is some electron configuration changes, right? So all those mechanisms are catalyzed by the enzymes. Okay, so basically यहाँ पे कुछ chemical reactions discuss किए हैं. So if we say breaking and formation of new bonds, then it may be hydrolysis. It may be just lysis in absence of water. Hydrolysis is in presence of water, right? Only lysis can be in absence of water. New bond formation, जैसे ATP बनता है तो एनर्जी रिच बॉन्ड वाटर के एब्सेंस में बनता है. ATP टूटता है तो वाटर के प्रेजेंस में उसका ब्रेक डाउन होता है. सो इट इज हाइड्रोलिसिस ऑफ ATP. So new bond formation or breaking, hydrolysis, only lysis, ligation, two chemicals joined together forming new bond. अब bond के प्रकार है, covalent bond formation है, right? Variety of things you have discussed in chemistry. Those are all catalyzed by chemical reactions. So these are the things. So let us read page number 154 in CRT, chemical reactions. How do we understand these enzymes? Let us first understand a chemical reaction. Chemical compounds undergo two types of changes. A physical change simply referred to change in shape without breaking of bonds. So you are having a metal, you are pressing it, you are changing the shape, its form changes, its shape changes, but chemistry remains the same. Okay, a physical change simply refers to change in shape without breaking of bonds. This is physical process. Another physical process is change in state of matter, like liquid to solid, solid to gases, like that. Okay, or solid to liquid, liquid to gases, like that. When ice melt into water or when water becomes a vapor. Okay, these are the physical changes. Again, the chemistry remains same. These are physical processes. However, when bonds are broken and new bonds are formed during transformation, this is called a chemical reaction. Okay, bond टूटता है, जुड़ता है, तो उसको हम बोलेंगे chemical reaction. For example, see it is given BaOH2 plus H2SO4 giving you BaSO4. So this SO4 group is taken out from H2CO4, your sulfuric acid, and added to the chemical, and water is given out. अब यहाँ पे कंडेंसेशन का रिएक्शन भी हमने अमाइनो एसिड्स में देखा है. टू मॉलिक्यूल्स कम टुगेदर, वाटर मॉलिक्यूल इज गिवन आउट, सो इट इज कंडेंसेशन. It can be opposite of condensation, that is cleavage, breakdown. Okay, see, in an inorganic chemical reaction, okay, so this above chemical reaction explained over here, all the chemical participants are inorganic. So this is inorganic chemical reaction. Similarly, hydrolysis of starch into glucose is an organic chemical reaction. Rate of a physical or chemical process refers to the amount of product formed per unit time. Okay, so this is what is rate of reaction. Okay, what is that? Rate of physical or chemical reaction refers to the product form per unit time. 1 minute में, 1 hour में, what number of product molecules are formed is rate of the chemical reaction, which is represented, rate of reaction is delta P upon delta T. Okay, initial final time, initial time में difference लेके कितना end product बनता है. So that is what is the rate can be calculated by this formula. Rate can also be called velocity if the direction is specified. You know two words, speed and velocity. You know scalar, vector, right? So velocity is a term which has unit as well as the direction. Forward velocity, reverse. Okay, so as we said, the chemical reactions are reversible. So that we are talking about velocity, forward and रिवर्सिबल. अब आप केमिस्ट्री में ये भी देखते हो, रेट ऑफ फॉरवर्ड केमिकल रिएक्शन इज के एंड रेट ऑफ रिवर्सिबल रिएक्शन इट विल बी K0, बराबर. सो K इज मोर देन K0, product will be more than substrate. Product will be more than substrate. If I said K is less than K0, then product is less than substrate. Okay, so ये सब आप केमिस्ट्री में सीख चुके हो. So that is what they are talking about, velocity. फॉरवर्ड केमिकल रिएक्शन का रेट ज्यादा है तो प्रोडक्ट ज्यादा रहेगा. रिवर्स केमिकल रिएक्शन का रेट ज्यादा है तो सबस्ट्रेट अंत में ज्यादा बनेगा, बराबर. सी, रेट ऑफ फिजिकल एंड केमिकल प्रोसेसेस आर इन्फ्लुएंस्ड बाय टेम्परेचर, among other factors. A general rule of thumb is that rate doubles on or decreases by half. Matlab, right now the rate is 10 molecules per second of substrate are formed. Okay, so just imagine at 20 degrees centigrade, rate of chemical reaction is 20 molecules of end product are formed in 1 hour. 20 degrees में 1 hour में 20 end product के molecule बनते हैं. If you rise the temperature by 20 degree to 30, तो ये रेट डबल होगा. तो ये रेट डबल होगा. 20 की जगह आपको 40 end product के molecules मिलेंगे. Okay, so this is what is called Q10, that is temperature coefficient. Okay, so that is what they are talking about. A general rule of thumb is that rate doubles or decreases by half for every 100 degree change in either direction, forward or reversible. Catalyzed reaction proceed at a rate vastly higher than that of uncatalyzed reaction. कैटलिस्ट प्रेजेंट है, एंजाइम प्रेजेंट है, तो केमिकल रिएक्शन का रेट बहुत ज्यादा होगा. When enzyme catalyzed reactions are observed, the rate could be vastly higher than the same but uncatalyzed reaction. For example, CO2 plus water, you get carbonic acid. Okay, it is a reversible reaction. Okay, so here they have given the speed. See, in absence of any enzyme, this reaction is very slow, with about 200 molecules of H2CO3 being formed in an hour. Okay, 1 घंटे में 200 carbonic acid बनेंगे. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction speed dramatically with about 6 lakhs molecule being formed. देखो, एंजाइम का मॉलिक्यूल आ गया तो रेट ऑफ रिएक्शन बहुत ज्यादा बढ़ती है. The enzyme has accelerated the reaction rate by about 10 million times. The power of enzyme is incredible indeed. So why this enzymatic reaction? Okay, देखो ना, it is 10 million time rate बढ़ गया. क्यों? एंजाइम है. तो ये पावर किसका है? एंजाइम की वजह से ये स्पीड बढ़ गया. There are thousands of types of enzyme, each catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction when each of the step is catalyzed by the same enzyme. Okay, there can be a series of multi-step chemical reaction which can be catalyzed by similar enzyme. The multi-step chemical reaction when each of the step is catalyzed by same enzyme complex or different enzymes is called metabolic pathway. जैसे anaerobic respiration में हम zymes complex बोलते हैं. Yeast anaerobic respiration involves diamase complex. It is group of enzyme. Okay, so series of chemical reaction में ये group of enzyme काम करेंगे and what you are getting, 1 glucose converted to pyruvic acid, right? Okay, glucose plus oxygen अगर aerobic respiration करते हैं then you are getting 2 pyruvic acid, 2 water molecules given out, or you are getting glucose plus oxygen giving you 2C3H4O3 plus 2 water molecules are given out. Okay, so enzyme complex are group of enzyme working together to cause one metabolic pathway. It is actually metabolic pathway in which glucose become pyruvic acid through 10 different enzyme catalyzed metabolic reaction. 10 ये number ध्यान में रखना, underline करना. When you study respiration, you will study this reaction. We have already studied, so no need to explain much. At the stage, you should know that this very metabolic pathway with one of 2 additional reaction gives rise to variety of metabolic end product. अब आप respiration पूरा देख चुके हो. Okay, respiration में हमने अलग अलग जंक्शन देखे, अलग अलग रॉ मटेरियल कैसे प्रोसेस होते हैं वो देखे, एंजाइम देखे. अब यहाँ पे ये पैराग्राफ समझना हमारे लिए आसान है क्योंकि हमने रेस्पिरेशन सीखा है. अदरवाइज दिस पैराग्राफ अगेन इज बाउंसर फॉर यू. ओके, सो प्लीज नोट, हमको ये चैप्टर पूरे लेसन से रिलेट करना है, इसलिए हमने ये बाद में कंक्लूड किया है. सी नाउ, इन आवर स्केलेटल मसल anaerobic respiration, under anaerobic condition lactic acid is formed. So you know glucose converted to pyruvic acid. Pyruvic acid is reduced to lactic acid. There is no decarboxylation. CO2 बाहर नहीं निकलता. Okay, so lactic acid is formed. Lactic acid accumulate causing cramp. Okay, fatigue muscle का. Under normal aerobic conditions, pyruvic acid is formed. In yeast, during fermentation, the same pathway leads to the production of ethanol. Hence, in different conditions, different products are possible. Okay, enzyme बदल गया, अलग-अलग chemical मेटाबोलाइट्स या फिर कोफैक्टर, जैसे मैं बोलती हूँ विटामिन मिनरल बदल गए तो एंड प्रोडक्ट बदलता है. Okay, so this is how variety of chemical reactions are catalyzed by enzyme, and while catalyzing it follows this property. Rate can be calculated by this delta P upon delta T, and if you rises or decreases the temperature by 10 degrees centigrade, the rate will be doubled up. If the temperature rises, rate will be reduced to half if the temperature is reduced by 10 degrees centigrade. Okay, so these are the basic properties or the way the enzyme work. Okay, अब ये जो properties है इसमें से कुछ property हमको आगे के लेसन के लिए relate करने हैं, तो उसको थोड़ा डिटेल में देखते हैं. See, now this is the overall process how enzymes work. Okay, so basic thing we'll discuss it and then relate with its property. This is the raw material A, raw material B. Raw material A, raw material B. I have purposefully drawn two different shapes. It is for like diagrammatic representation, and you know each chemical has different structural formula, different chemical formula. So shape means different chemicals. So A raw material, B raw material. They are actually stable, will not react with each other if this enzyme is not there. Or if at all they react, then the rate will be very slow. See, each chemical has kinetic energy. Okay, kinetic energy, if we are talking about within a cell, it may be because of cytoplasmic streaming. You know, cytoplasm continuously rotates. It may be because of the kinetic energy which is dependent on the temperature. So accidentally, the raw materials may colloid. Okay, you know, more the collisions, more will be the rate of reaction. ये पूरा chemistry का knowledge यहाँ पे आप use कर सकते हो. So these are the raw material randomly meeting each other, and this chemical reaction may happen. But if enzyme is present with particular active sites, again, shape of the active site is representing the specificity. Okay, here particularly we are using complementary shapes for recognizing the active sites, right? So A के लिए ये V-shaped groove is the active site, और B के लिए ये rectangular shaped groove is the active site. So enzyme and raw material accidentally meet. Now this enzyme recognizes its own raw material. Okay, once it is recognized, that chemistry is analyzed, orientation is even important, and then the enzyme loosely bound to the raw material recognizes it, and then it tightly bound with it by either changing its shape slightly, okay, changing in configuration, right, which is a 3-dimensional. So it will recognize, and now the enzyme is with the raw material. So इसको हम बोलेंगे, it is enzyme substrate complex. Enzyme substrate complex. Because of this complex, now the raw material is activated, and activation of energy required for these A and B in presence of enzyme is very less. Okay, so this activated A and B chemical now start reacting with each other and produce a end product, that is AB. Okay, so AB when attached, अब यहाँ पे देखो chemical reaction हो गया, इनके बीच में bond बन गया, तो अब हमको मिलेगा enzyme product complex. This is a transition step. ये एक transition step है. Okay, जिसमें enzyme substrate complex converted to enzyme product complex, and the product is set free. And the product is set free. Okay, product निकल गया, enzyme is recycled. So this is how the chemical reaction always happens. Okay, so चलो ये काम property के साथ देखते हैं. It has 3D conformation. It has specific structure because of which it has those active sites, and it can recognize its own chemical. It has one or more active sites. See, I have drawn here 2 active sites. अब यहाँ पे मैंने एक बात बोली थी कि ये V-shaped groove A chemical के लिए active site है, तो क्या ये किसी और केमिकल के लिए एक्टिव साइट हो ही नहीं सकता? हो सकता है. जस्ट इमेजिन दैट देयर इज अ केमिकल विथ दिस शेप. Okay, I am labeling it P. It is a poisonous chemical. So see, poison is also having orientation or structural similarity, complementary nature with the active site. So for this active site, P and A compete with each other. For this active site, P and A compete with each other. अगर A का concentration ज्यादा है, तो enzyme react with A, chances will be more. If concentration of P is more, enzyme reacting with P, the possibility is more. Right? So, sometime what happens? Poisons come and bind with the enzyme permanently. अगर ये P poison A site के यहाँ पे लोकेशन पे बाइंड हो गया परमानेंटली, तो ये एंजाइम मॉलिक्यूल स्ट्रक्चरली कंप्लीटली ट्रांसफॉर्म्ड हो गया और ये नॉन एक्टिव फॉर्म में चला गया. जस्ट इमेजिन, if poison make this enzyme inactive, this chemical reaction will stop and there will not be AB product. And if AB product is life essential, the cell will be dead in presence of this enzyme. समझ रहे हो? So it has one or more active site. One active site is sensitive to one or few chemicals. Okay, A compete with P for this particular site. So ये दोनों के बीच में competition है ये site के लिए. Then enzymes are recycled. See, at the end of chemical reaction, enzymes are recycled. Then it is specific in action. This enzyme will react with A and B at particular temperature, particular pH. So it is action specific. Very sensitive to pH and temperature. All these processes are happening requiring particular pH, particular temperature. It is reversible. ये प्रोसेस रिवर्सिबल हो सकता है. अब यहाँ पे मैंने क्या बोला था? दिस इज अ फॉरवर्ड केमिकल रिएक्शन. फॉरवर्ड केमिकल रिएक्शन. ओके, सो सी, फॉरवर्ड केमिकल रिएक्शन, इसके लिए अगर हमने बोला K2K0, ओके, और रिवर्स केमिकल रिएक्शन K0 to K. Okay, so these are reversible reaction. This will happen when more raw material is available, so forward chemical reaction का रेट ज्यादा रहेगा. Okay, यहाँ पे more end product, तो reversible chemical reaction का रेट बढ़ सकता है. This is one parameter. Or might be ये chemical reaction 4 pH पे हो रहा है, तो ये केमिकल रिएक्शन 5 pH पे होगा. Okay, so this is what is reversible reaction, and enzyme reduces activation energy required for activation of raw material. So this is what is enzyme biocatalyst. You can relate its activity with catalyst, and you have to relate this with your chemistry. Mechanism of enzyme action. Let's help. Properties of enzymes we have discussed. Now, how it actually works. Now, different scientists propose different methods. Among these two, the first one was lock and key model, and the other one is induced fit hypothesis. Okay, we'll explain it as diagram. लेकिन थोड़ा शब्द की तरफ अगर ध्यान देते हैं, तो see, lock is the enzyme and substrate is the key. For particular lock, there is particular key. So, हर lock, that is enzyme के लिए एक key है substrate जो उसको पहचानती है. Right? So, enzyme is a lock, substrate is the key. Key recognizes its own lock and get fit. Okay, अब यहाँ पे lock and key को अगर हम compare करते हैं, तो lock and key में दोनों के shape fix है, complementary है. They won't change. So each enzyme is specific. It will work only with one key. So एक enzyme के लिए एक ही substrate बना है, वो उसके लिए fix है, उसके शेप में कोई बदलाव नहीं होता, तो स्पेसिफिसिटी बहुत ज्यादा प्रिसाइज है और मेंटेन होती है. Okay, so that is what both have complementary and fixed shape. It is an old concept, right? And the other one is induced fit. इसमें enzyme has particular shape, active site, but it is a flexible molecule. It goes on wandering in the cytoplasm. It goes out of the cell, search for the substrate. Accidentally, substrate and enzyme meet. Okay, as the molecules are continuously moving, cytoplasmic streaming is there. Molecule has kinetic energy, it is dependent on temperature. That's all that. Because of that, enzyme and substrate meet. When enzyme come across the substrate, it hold it, judge it, recognize it, and accordingly it modifies itself. Okay, so enzyme खुद की active site modify कर सकता है. Enzyme खुद की site modify कर सकता है. So that is what is induced fit. When the substrate approaches the enzyme, then the enzyme recognizes it and it can modify itself. It may go for conformational changes. Right? उसके structure में, उसके काम करने के तरीके में conformational changes होंगे. उसकी वजह से वो अपने substrate को पहचान लेगा. वो अपने substrate को पहचान लेगा. So enzyme is flexible and its active site changes depending on the substrate, depending on the cofactor, depending on the coenzymes, depending on variety of things, and then it will follow this method of reaction. Okay, so देखो अब reaction कैसा होता है? What is the method of reaction in cell cytoplasm? Anywhere. See, enzyme is there in the cytoplasm or outside the cell. There is substrate. More the substrate, more will be the chances of meeting enzyme with the substrate. Okay, so enzyme and substrate recognizes itself. Conformational changes will be there. Enzyme holds the substrate and now they form a complex. So that is enzyme substrate complex in which the chemicals, the substrate, raw material are activated. Okay, in presence of enzyme, energy of activation which is required for a substrate reduces drastically. अगर एंजाइम नहीं है और आप चाहते हो कि केमिकल रिएक्शन में पार्टिसिपेट करे, तो आपको उसको एक्टिवेट करने के लिए जो एनर्जी लगती है उसका अमाउंट अगर हम एंजाइम के प्रेजेंस में लगे वाले एनर्जी से कंपेयर करेंगे तो देयर इज ह्यूज डिफरेंस. सो एंजाइम रिड्यूसेस एनर्जी ऑफ एक्टिवेशन फॉर दैट पर्टिकुलर सबस्ट्रेट, राइट? सो सी, एंजाइम complex form, enzyme activate the substrate, enzyme reduces the activation energy, enzyme makes the raw material imbalanced active so that it participate in the chemical reaction. Okay, so enzyme participate in the chemical reaction and ultimately after this process the product is formed and now the enzyme is holding the product. It is called EP complex, enzyme product complex. This EP complex will be there together as the stable chemical product is formed, the product is released and enzyme is recycled. Okay, so these two hypotheses, induced fit and lock key, will discuss with the help of a diagram. अब ये शब्द NCERT में है लेकिन डायग्राम्स के आधार पे हम उसको बहुत अच्छे से समझ पाएंगे. So for better understanding, let us discuss these two diagrams. See the diagram sequentially. The yellow globular structure is enzyme and the upper pointed structure is the substrate. Now substrate and enzyme are coming in contact with each other, right? Because it recognizes, active site recognizes that particular substrate. Active site recognizes that particular substrate and the substrate come and bind to the active site. See, the shape of the substrate and the enzyme are completely complementary and they are fixed structures. So enzyme plus substrate react with each other. You get enzyme substrate complex wherein the enzyme activates the raw material, reduces its activation energy, and now this activated raw material undergo और पार्टिसिपेट इन केमिकल चेंजेस. बॉन्ड फॉर्मेशन होगा, एक्सचेंज होगा, कुछ बॉन्ड ब्रेक होंगे, कुछ बॉन्ड नए बनेंगे, राइट? So accordingly the chemical reaction happens, and after catalyzing the chemical reaction you can see enzyme product complex in which the enzyme is as it is, but the substrate is broken down into two parts. One is blue circle and the other one is green pointed part. So initially one chemical, right, just imagine जो substrate है वो chemical का नाम है AB, वो substrate का नाम है AB. अब AB टूट गया तो हमको A chemical अलग मिलेगा और B chemical अलग मिलेगा. Are you getting the point? What I said? This is AB substrate coming in contact with the enzyme, fix it. Right? It forms enzyme substrate complex. This activates, and now the enzyme produced break down these two chemicals, and you get the chemical A and chemical B separate but still attached to the enzyme. So it is enzyme product complex. Now the products are released. Green part is separated as well as the blue part is separated. So it is like a lysis of the chemical. AB chemical lies to produce A and B, and enzyme is set free. Enzyme will be recycled. This hypothesis is called lock and key hypothesis. See this one. Enzyme doesn't have fixed active sites. They are flexible. It is a flexible molecule. Yellow and blue are the substrate. Accidentally made the enzyme. Okay, it recognizes the substrate, and accordingly the enzyme modify itself. It undergo conformational changes, and now both the substrates are bound to the enzyme, and you get enzyme substrate complex. And now these two chemicals being active, they react with each other. Yellow one react with the blue one, and the product is formed. Okay, so you get enzyme product complex, product is released, and enzymes are recycled. So this is induced fit model, which is widely accepted, which explains the structure of active site, which is flexible. It goes on changing as reference to coenzyme कौन सा है, cofactor कौन सा है, raw material कौन सा है, pH क्या है. Okay, so as protein molecule structure is dependent of variety of things, exactly same concept will be describing that the active site of the enzyme goes on changing. So this is induced fit hypothesis. NCERT रीडिंग में केमिकल रिएक्शन, यही सब अब हमको एक्सप्लेन करना है. सी, आई एम ऑन पेज नंबर 154, केमिकल रिएक्शन. हाउ डू वी अंडरस्टैंड दीज एंजाइम्स? Let us first understand a chemical reaction. Chemical compound undergo two type of changes. A physical change simply refer to change in shape without breaking of bond. जैसे solid से liquid, liquid से solid या gaseous stage. Okay, just shape change होता है, लेकिन chemical nature same रहता है. That is physical change. This is a physical process. Another physical process is change in state of matter. When ice melt into water or when water become a vapor. There are physical processes. Okay, so physical change में gaseous state to liquid state, liquid to solid ये होगा, या आप mold करके उसका shape में बदलाव करेंगे. Okay, still the chemical configuration remains same. These are physical processes. However, when bonds are broken and new bonds are formed during transformation, this will be called a chemical reaction. But when the nature of bond changes, the atoms participating to form particular molecule, its combination changes, right? Then it will be a different chemical, and it won't be just a chemical, it won't be just a physical change. See, for example, BaOH2 plus H2SO4 gives you BaSO4 plus 2 water molecules. यहाँ पे H2SO4 का SO4 group is now added in the element Ba, and now you are getting a water molecule. So this is a chemical reaction of dehydration. See, we'll talk about this. This will be called chemical reaction. For example, just now we go through it. In an inorganic chemical reaction, similarly hydrolysis of starch into glucose is an organic chemical reaction. इसमें ये chemical जिन्होंने participate किए inorganic थे. Next chemical reaction में भी breakdown ही हो रहा है but organic chemical का. What is that? Starch is broken down into glucose. Rate of physical or chemical process refers to amount of product per unit time. More the rate of chemical reaction, more will be the product form. It can be expressed rate delta P upon delta T. Okay, amount of product produced per unit time determines the rate. This is rate of the chemical reaction. Rate can also be called velocity if the direction is specified. You know two words, speed and velocity. Okay, velocity, okay, vector, scalar, physics में सीखा है ना. Velocity is speed in one particular direction, so it has unit as well as the direction. Okay, so see, rate can also be called velocity if the direction is specified. Forward chemical reaction, backward chemical reaction. Rates of physical and chemical processes are influenced by temperature among other factors. Okay, so it is dependent on temperature. A general rule of thumb is that rate doubles or decreases by half for every 10 degrees centigrade change in either direction. Okay, so for example, if we say that 10 enzymes are reacting with 100 substrate, 10 enzymes are reacting with 100 substrate, and you are getting 10 end product per unit time. But if you increase the temperature by 10 degrees, then your product, instead of getting 10, you will get 20. So production of product will be doubled up after rise in temperature. और उल्टा अगर करते हो, टेम्परेचर 10 डिग्री से कम करते हो, तो प्रोडक्ट फॉर्मेशन का रेट हाफ से कम होगा. Right? So what is that? Is in either direction. I repeat, see, a general rule of thumb is that rate doubles or decreases by half for every 10 degree changes. It's also called Q10. Catalyzed reactions proceed at a rate vastly higher than that of uncatalyzed reaction. If enzymes are participating, catalysts are present, rate of reaction is very fast. If it is less, then the rate of reaction is low. When enzyme catalyzed reaction are observed, the rate would be vastly higher than the same but uncatalyzed reaction. For example, carbon dioxide plus water giving you carbonic acid. It is a very common process. Even in previous lecture we talk about that. Okay, so absence of enzyme 200 mole per hour. Okay, अब यही chemical reaction अगर enzyme नहीं है तो 200 mole per hour. Enzyme है तो it will be 6 lakhs mole per hour. Okay, so that much power the enzyme has. In absence of enzyme, this reaction is very slow with about 200 molecule of H2CO3 being formed in an hour. However, by using enzyme presence within the cytoplasm called carbonic anhydrase, the reaction speeds up dramatically with about 6 lakhs molecule being formed every second. Right? So see, the enzyme has accelerated the reaction rate. यहाँ पे second है, R नहीं है, please ध्यान में रखो. The enzyme has accelerated the reaction rate by about 10 million times. This power of enzyme is incredible indeed. There are thousands of types of enzyme, each catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction, when each of the steps is catalyzed by the same enzyme complex or different complexes, is called metabolic pathway. Okay. So, series of chemical reaction catalyzed by one enzyme or variety of different enzymes. So that series of chemical reaction is called pathway. It may be linear if it is pathway, circular if it is cycle. Glucose to pyruvic acid is pathway. Is actually metabolic pathway in which glucose becomes pyruvic acid through 10 different enzyme catalyzed metabolic reactions. When you study respiration chapter 14, you will study these reactions. Okay, हमने पहले respiration सीखा, अब हम ये पढ़ रहे हैं. Okay, so that's why the statement is कि ये reaction आप respiration में सीखोगे, तो हम सीख चुके because we altered the sequence, right? Okay. At this stage, you should know that this is very metabolic pathway with one or two additional reactions giving rise to variety of metabolic and end products. In our skeletal muscle, under anaerobic condition lactic acid is formed. Okay, पूरा respiration lesson याद करो. Okay, first step glycolysis gives you 2 molecules of pyruvic acid. Pyruvic acid is reduced to lactic acid. There is no decarboxylation in anaerobic respiration of muscle cell. Under normal aerobic condition, pyruvic acid forms and it follows acetylation TCA, and all reduced coenzymes follow electron transport system. In yeast, during fermentation, the same pathway leads to production of ethanol. Okay, हमने ये देखा भी था, common pathway glycolysis. Glycolysis participate in aerobic respiration, 38 ATP are formed. Right? Glycolysis participating anaerobic respiration. Glycolysis के आगे anaerobic respiration हो रहा है. Then it will be forming 2 ATP, 2 molecules of ethyl alcohol. Okay, in fungus or in plant cell. If it is anaerobic respiration of animal cell, it produces 2 molecules of lactic acid, 2 ATP, no decarboxylation. आप ये पूरा सीख चुके हो. Aerobic respiration as well as 2 types of fermentation processes. Hence, the different condition, different products are possible. Okay, how do enzymes bring about such high rate of chemical convergence? To understand this, we should study enzyme a little more. Okay, ये पूरा इतना mechanism कैसे होता है वो देखते हैं. When we already understood the idea of active site, the chemical or metabolic conversion refers to reaction. The chemical which is converted into a product is called substrate. Hence, enzyme, that is protein, with their dimensional structure including an active site, convert a substrate into a product, capital P. Symbolically, this can be depicted like this. Okay, so see S, that is substrate converted to product in presence of enzyme. It is now understood that the substrate S has to bind to enzyme at its active site within a given cleft or pocket. Okay, अभी हमने दोनों मेथड देखे, induced fit as well as lock and key. The substrate has to diffuse towards the active site. There is thus an obligatory formation of ES, enzyme substrate complex. Right? E stand for enzyme. This complex formation is a transient phenomenon. It is a periodic, transitional. Okay, ये complex बनेगा, readily you will get this one. Reaction के लिए इतने कितना टाइम लगेगा वो डिपेंड करता है chemical reaction पे, okay, और raw material temperature पे. Okay, so this is a transient state, short term process. During the state where substrate is bound to the enzyme active site, a new structure of the substrate called transition state structure is formed. Very soon after a expected bond breaking, making is completed. Okay, ये complex बनेगा, raw material activate होगा, chemical reaction में participate करेगा, bond तोड़ के उसको active किया जाएगा, नए bond बना के product बनाया जाएगा. Okay, the product is released from the active site. In other words, the structure of substrate gets transformed into structure of product. Raw material substrate is transformed to product. Product is released and enzyme is recycled. The pathway for this transformation must go through the so-called transition state structure. There could be many more altered structure states between the stable substrate and the product. Okay, इसके बीच में stable substrate activate हो रहा है. Okay, फिर उसको bond तोड़ के chemical reaction में खींच के उतारा जा रहा है और ultimately हमको product मिल रहा है. So it is all dependent. Implicit in this statement is the fact that all other intermediate structure states are unstable. Stability is something related to energy status of the molecule or the structure. See the graph 9.6, concept of activation energy. Okay, अब ये पूरा bond formation, chemical reaction, okay, reaction का energetics, okay, thermodynamics, आप पूरे chemistry में सीखते हो. ये stable chemical बनने के बाद energy कम होती है, ये आप chemistry में भी सीखते हो. Okay, so see, it is radiant energy on the y-axis, x-axis, progress of reaction. Okay, substrate. In presence of enzyme, in absence of enzyme. Okay, see, activation energy without enzyme. See, how much high, more than 3 times energy of activation is required in absence of enzyme? It is 1/3. An activated chemical readily reacts with each other and you get a product with release of energy. जैसे ही स्टेबल केमिकल बनेगा एनर्जी रिलीज होगी. ओके, ये नॉर्मल इनऑर्गेनिक केमिकल रिएक्शंस की हम बात करते हैं. फोटोसिंथेसिस में ग्लूकोज बनता है तो एनर्जी एब्जॉर्ब होती है बिकॉज इट इज हैविंग एनर्जी रिच केमिकल. ओके, ग्लूकोज इज अ एनर्जी रिच केमिकल. ओके, सो यहाँ पे थोड़ा हमको डिफरेंस देखना पड़ेगा. ये ऑर्गेनिक एनर्जी रिच केमिकल बन रहा है तो एनर्जी एब्जॉर्ब होगी, राइट. एक स्टेबल इनऑर्गेनिक केमिकल बन रहा है तो एनर्जी रिलीज होगी. ओके, सो सी, stability is something related to energy status of a molecule or the structure. Hence, when we look at this priority through a graph, it looks like something as figure 9.6. The y-axis represents potential energy contained. The x-axis represents the progression of structural transformation or state through which transition state. Okay, जहाँ पे changes हो रहे हैं. You would notice two things. The energy level difference between S and P. If P is at a lower level of S, the reaction is exothermic, just now as I said. Okay, one need to supply energy by heating in spontaneous in order to form the end product. However, whether it is an exothermic or spontaneous reaction or endothermic or energy requiring reaction. देखिए ये डिपेंड करता है. एनर्जी अब्जॉर्ब होगी अगर एंडोथर्मिक रिएक्शन है, एनर्जी रिलीज होगी अगर एक्सोथर्मिक रिएक्शन है, राइट. सी, द डिफरेंस इन एवरेज एनर्जी कांस्टेंट कंटेंट ऑफ एस सबस्ट्रेट from that of its transition state is called activation energy. Ground state से excited state तक, okay, केमिकल को एक्टिवेट कर दे, केमिकल रिएक्शन में उतारने की क्षमता एक्टिवेशन एनर्जी करती है. Enzyme eventually bring down this energy barrier, making the transition of S to P more easy. Okay, because of enzyme, activation energy is reduced and conversion of raw material to product is easy in presence of biocatalyst, that is enzyme.